A quick revision about Staphylococcus aureus. Staphylococcus aureus is gram-positive coccus and it is arranged in clusters, and it is look like grape-like cluster. It is catalase positive, coagulase positive, non-motile, non-sporing, and occasionally capsulated. Then about its virulence factors. The important virulence factors include teichoic acid, capsular polysaccharide, protein A clumping factor, and coagulase, also C-reactive proteins. Teichoic acid helps in adhesion to mucosal surface and it inhibits opsonization. Then capsular polysaccharide. We know that gram-positive bacteria have a thicker cell wall or peptidoglycan layer. It confers rigidity to cell wall and maintains the shape. Then protein A. It is encoded by SPA gene and it has many biological properties like anti-complementary action, chemotactic or mitogenic. Then it inhibits opsonization and induces platelet damage. Protein A also mediates coagglutination reaction. Then about clumping factor. It is a surface protein and it is responsible for slight agglutination test. Then coagulase. It is responsible for clotting of plasma. Then C-reactive protein. It is a plasma protein which activates prothrombin, which convert the fibrinogen to fibrin. Then about toxins. The toxins include cytolytic toxin, epidermolytic toxin, enterotoxin F, enterotoxin type A. First about cytolytic toxin. It includes alpha hemolysin, beta hemolysin, gamma hemolysin, then delta hemolysin. Also pattern valentine toxin or leucocidins. Then epidermolytic toxin. It is also called exfoliate toxin. And there are ET1 and ET2. Two types of toxins are included in epidermolytic toxin and which is responsible for staphylococcal scalded skin syndrome. Then about enterotoxin F, which is responsible for toxic shock syndrome and in case the drug of choice is clindamycin. Then about enterotoxin type A, which is responsible for food poisoning. There are also some other type of enterotoxins are present, but important is type A enterotoxin. Here the treatment is rehydration and correction of the electrolyte balance. Then about clinical features. Here the mode of entry of bacteria is through skin abrasions or mucous membrane trauma. And the infections include hospital acute infections, skin and soft tissue infections, osteomyelitis, food poisoning, toxic shock syndrome, abscess, etc. Then about lab diagnosis. Here the microscopic part is explained in the first slide. We are directly going to the culture. The culture media include nutrient agar. In nutrient agar we get golden white colonies with a oil paint appearance. In the figure we can see golden white colonies with the oil paint appearance. Then in blood agar we get beta hemolytic colonies. We can see the appearance of Staphylococcus aureus in the blood agar. On mechanical agar we get lactose fermenting colonies. Then about selective media. The selective media include mannitol salt agar, salt melt agar, and Lutlam's media. In mannitol salt agar we get mannitol fermenting Staphylococcus aureus colonies. Then salt melt agar. It contains nutrient agar, 6.5% sodium chloride and 10% skimmed milk. And in Lutlam's medium contains lithium chloride and tellurite. Then biochemical reactions. Staph aureus is catalase and urease positive. Medial red is also positive. Then coagulase test. It is most commonly used biochemical reaction for identification of Staph aureus. There are two types of coagulase test, tube coagulase test and slide coagulase test. The tube coagulase test is mediated by the protein A and the slide coagulase test is mediated by clumping factor or bound coagulase. In the figure we can see tube coagulase test. Here the test tube A shows tube coagulase positive result and B show tube coagulase test negative, and it is the slide coagulase test. Here the positive result, here the negative result. Then alteration penicillin binding protein detected in genotyping and phenotyping. And the typing method include phenotypic method like bacteriophage typing and antibiogram typing. And the genotypic method include PCR-RF-LP. Then about treatment. Since Staph aureus rapidly develops drug resistance, the antibiotic should be cautiously chosen. The sensitive to penicillin strains, the drug of choice is penicillin G or dicloxacillin, and sensitive to methicillin cases, the drug of choice is neficillin or oxacillin. Then MRSA, the drug of choice is vancomycin, linezolid, etc. MRSA means methicillin resistant Staph aureus, and in VRSA, that is vancomycin resistant Staph aureus, the drug of choice is 5th generation cephalosporins like ceftobiprole, ceftibuten, etc. The MRSA resistant Staph aureus is mediated by chromosomally coded gene cat MECA gene, which alter the penicillin binding protein PBP present on Staph aureus cell membrane to PBP2A. Then VRSA, it is mediated by VAN-A gene. The VAN-A gene is believed to be acquired from the vancomycin resistant strain of Enterococcus faecalis by horizontal conjugal transfer. So it is only a quick revision of Staph aureus topic.